Okay, any announcements? Yeah, lab practicals, right? You guys had it yesterday. Yes, I graded y'all's lab practicals and I uploaded the scores. Was that you? Yeah, it was. Monday, Wednesday. Yeah, you guys are, yeah, that's right. So I uploaded the scores. Take a look at those. Obviously, you can't see. Now I'm doubting myself. Did you take a lab practical yesterday, Marie? Yes? Yes. See, the problem is, here's what's happening. As the end of the semester comes close, both my full semester Bio 2 and my mini semester Bio 2 start merging, right? So toward the end, I'm like, oh, we're both doing the same thing, and that really makes it worse. And anyway, so I posted the grades. It's out of 150. You can't see the practical, obviously, but of course, I'd be more than happy to show it to you along with the questions. Um, I was going to say that's the best way for me to go over it with you. Going over it with it online is difficult because I can show you the questions, but I guess I could read your answer. It's up to you, but probably the best way is to look at it in lab, face to face. All right. So where were we in the plants? We were in the plants that are the container plants. That's what the word angio means. Angio is a Latin or something or other word. I think it's probably Latin. It means container. And the container we're talking about is that very important container that angiosperms have. You see, all along, since the beginning of the semester, but certainly within the plants, we've been talking about how do plants change their way to deal with different environments. Either the environment is changing, or of course, like we've talked about, if you can do it, if you can hack it, go into an environment where there's very few or anybody else because you get all the resources. So as we worked our way up into the plants and we get into the angiosperms, we have these two big things that stand out about angiosperms. Now, there's a couple of other smaller things that give them an advantage we'll go into, but the big thing, of course, is, like I said in this picture, is the fact they have flowers. Remember, plants don't make flowers they don't make pretty flowers because, oh gosh, they want to. They make them because they're trying to attract pollinators, right? Unless they're small flowers. Small ones just usually use wind, or I don't know, I guess maybe some small flowers could use insects. I don't see why not. Anything that crawls from flower to flower could be a pollinator, right? But in this case, the container part is really important too, because the seed, which we saw back in the gymnosperms, the first time we saw it, remember, okay, try to get that square in your head. So we go from spores and mosses, spores and ferns. We still have spores here, but now we have seeds, right? Seed plants. But now we got seed plants with flowers and fruit. The seed is the important thing. It's the reproductive part. It's the thing you're trying to disperse. You're trying to disperse it for the reasons we've talked about all semester long. Where did you go, Cooper? For reasons that we've talked about all semester long, you're trying to disperse. It's better to get away from your parents, okay? We've got all of that. One of the ways you can do it is provide something for them to eat. Now, this is all going to happen physically in the female part of the flower. Whether you have male or female parts, everything that I'm describing, this creation of the fruit, is because, I don't have my pencil, is because the fact that the ovary right here, have it on, there we go. The ovary right there, that's going to become the fruit. Give me 1 second. So you can still hear me, hopefully, but it's the ovary that becomes the fruit. So when you eat tomato or, anything that's got seed, then that is the fruit, or that is the ovary. Even in a cucumber, which has seeds that we call, obviously, a fruit. Oh, I don't have my pencil. I do have my pencil. Where is it? Forget it. Anyway, that's what we mean. That's physically where all this is going on that I've been talking about, where this seed is located. Seed or ovule. Remember what I said in class. Don't get too hung up on the difference between ovule and seed. Let me sign Cooper out because something's going wrong here. Anyway, when does an ovule become a seed? It's kind of like what is embryo fetus, or is it fetus embryo? Embryo then fetus. When does that happen in humans? But anyway, eventually a seed or seeds are actually going to form this fruit thing, okay? So, and again, let me run through the parts of the flower. There are male and female parts on most flowers, I believe most flowers, although there are some flowers that exhibit what we would call sexual dimorphism. So what we mean by that in sexual dimorphism is that when we look at organisms, there is a male and a female, okay? So sexual dimorphism not only means that something is dioecious or gonorchid, are the words I've used, which mean that there's males and females in separate things. Remember cycads, the plants, the cones plants, there's a male one and a female one, right? So they're dioecious, but the pine trees that have both cones, pollen, male, female, ovulate further down, that's a monoecious plant. So it's kind of the same thing we're talking about here in flowers. Now, if a flower both has the male and female parts, it's complete. But now in the second one, it's incomplete. This could be in separate flowers, same plant, or separate plants, right? These are what we call incomplete flowers, because they either have like the ovary in the left or the stamen. All right, so stamen is the male part. Anthers are going to produce the pollen. Pollen travels down the style, attracted to it by the stigma. I was cutting some plants open the other day, and actually, if you take your little finger to touch this, you can feel it's a little bit sticky. Anyway, the pollen makes its way down there, and we'll talk about that in just a second. And eventually, it's going to fertilize the seed, okay? Parts. We'll do this in lab too. Sepals, petals, and the male parts, the female parts. I want you to know the naughty bits of the flower, obviously. Flowers are things that make plants attractive, whether you do it through normal light, whatever color you use, whether you're attracting insects or just just the wind. It's the way you disperse yourself. It might be the way you disperse pollen. It's going to be it's going to be the way you disperse pollen. It's also going to be the way you disperse, fertilized seeds, youngin. All right, now you can do it by tricking insects. Either get in and crawl on you, get a little bit of nectar, but touching them with those stamens, which of course have those anthers. Boom, there you go. Remember it wasn't you. Shoot, that's happened. See? It was the other class. We were talking about how insects have little hairy parts on them. Well, pollen sticks to all that. You can see it in the wasp. Of course, the Ophrys flower, yes, is trying to mimic a female for obvious reasons. All right, now, this is where we stopped. The carpel. I know these are new terms, so I do like to go back and remind yourself or remind everybody where we are. The carpel. Up here in the upper right. What did I do with my pen? Anyway, that's the female, sometimes called the pistil. I don't like the word pistil as much because to me it messes me up visually. Because when I think pistil, I think of a pistol, and that makes me think of penis, and that doesn't help me, right? Just a visual thing. So I'm glad that pistil is kind of an older word, but if you see it, I don't think your book uses it. I don't think. They might. But anyway, carpel, the female part of the plant, okay? The female part of the plant is, let me go ahead on the slides where we are. The female part of the plant, where all this is going to occur, is a modified leaf. Now, I've said this before, okay? All parts of the flower are modified leaves. The sepal are modified leaves, even though they kind of even look like leaves. Remember, they're the green things right underneath the petals. The petals, orange and all the weird colors and the cool colors that you get, those are modified leaves. The anther, the stamen, the filament, the male parts, modified leaves. Now, look, here's an example of where we see in everyday life, things you've probably eaten if you eat them, where we see an example of what I'm talking about. This is what we call split peas, I guess. I remember my mother sitting in the kitchen and breaking the tops off of these things. I'm getting ready to use the real pen. I better put that away. But, snapping these off. Not split peas. What did she call them? Snap beans? Anyway, I don't recall. They were pretty good. Now here, let's think for a second. Let's kind of go back in time and imagine a scenario by which we could get this fruit. Because that pea is a fruit, right? The little things inside, the peas inside, the green things inside are ovules. See how they're labeled in the last picture? They're the seeds. Now there's a picture up above to show you, if you've ever looked inside one of these. Reminded myself. I got to bring these to class. Hold on one second. I always do this lecture, and then we go to class, and I'm like, doggone it, I need beans. Peas, I need to bring those to lab so we can do this, right? Those are the seeds. Those are there's the carpel, right? The carpel being the container thing, the stigma, the style. I mean, it's obviously, in many cases, the style is going to fall off, so you get the bottom part, the ovary, where the fruit is. This is a fruit. It has seeds, okay? All right, go backwards in time. Let's go to the left now. What if what we imagine those seeds in that split pea, what if we unsplit the pea? Getting something that tells me that you're having troubles hearing or seeing me. Is that true? Can you confirm that for me, Marie? Because I can turn my video off if that's the case. Okay. All right, so I'll continue on. So, you know, if we go back and we take the pea, and this is why I don't remember bringing them to lab. So if we were to, you know, cut, or you can use your fingernail actually, to split right along there, you can open it up. Now, imagine that thing fully opened up. The picture that's totally on the left, right? So imagine that what that is that's a modified leaf, because when you open that pea up, it looks better in the picture than it does in lab. You got to pick the right pea, to be honest. But it looks like a leaf, doesn't it, in that picture on the left? And look what's hanging off of those. Seeds, ovules. So if we kind of apply evolution to this, we could say ovules, seeds, juniors, youngin, which you want to protect, right? These are plants that we call, you know, embryophytes. These are plants that are actually nurturing their seeds. They're actually doing something about it. They're not like the spores and the gametes back in the mosses. So anyway, through evolution, there would be an advantage, right, to closing that up, wouldn't there be? There would be an advantage to protecting the seeds, wouldn't there? To closing it up, to forming the carpel all the way around it, okay? Because remember, the carpel, anyway, this is all the carpel, right? So let me go back again and reinforce where we're talking about in the plant. We are right here. So as I said, oftentimes what happens is once the seeds are fertilized, this falls off. You don't need this anymore, right? That's what fruits are roundish, right? Aren't they? Let me think. Yeah, I don't know any square fruits. But anyway, for the most part, they are. All right, so you get the idea? So this is obviously just how it happened in these, but this is kind of a plausible hypothesis, don't you think? This is what I always try to reinforce that in this idea of form and function. It's because it helps you remember as the concepts get a little more in depth as you go through school. You always keep in mind what it looks like, how it works, and how that fits into the overall equation, right? You're pretty good at doing that, so make sure that when you study, you do that, okay? So anyway, however that happens, and it would be different, you know, of course, in different fruits. When you thicken the ovary wall, when you thicken the wall of that carpel, that round thing, maybe the style fell off, you form the fruit, which does those two things that I've already talked about. Now, depending on how you Lamarck, depending on how you have dispersed, depending on how you've adapted to dispersal, finally, you're going to do it like maybe a tomato that has seeds, so something is eating it. You know, oh, it's good. Dropping seeds. Grapefruits, there's another one. Nectarines, those are good to eat. Now, they produce a pit. That thing in the middle is a pit, but that is their seed. It's pretty hardy, you know. So now, seeds basically, maybe not all seeds, but most seeds I know, the way that they germinate is when water flows into them. You see, the way seeds are made is they're hypotonic. That's why you got to add water to them, folks. When you put it in the ground, you got to add water. Why do I have to add water? It's not a plant yet. Well, no. One of the things that the water does is, of course, provide water for survival. But since the seed is hypotonic, when you pour water on it, water goes into it, doesn't it? Right? Which is what you want to happen. So water does flow into the pit. Nuts or seeds, the inside anyway, and you can see that's a nice protective coating. What kind of seed dispersers? Now, these wouldn't be pollinators, remember. Don't confuse that. Pollinators transfer pollen. But what kind of seed dispersers do you think this, what is that, milkweed? Oh, I think I'm allergic to that. These are the seeds right here. It's kind of like cotton. If you take a raw cotton and you open it, we'll try to do that in class. I have some. You might be able to find the seeds in there. But what kind of dispersal method do you think milkweed or cotton uses? Boy, I'll bet. Yeah, cotton, or maybe some of them actually use, the fact that, fibers can cling together. Yeah, but for the most part, I would imagine it's wind that is the pollinator there, right? So you probably don't find very big flowers on those things either. I don't know what a cotton flower looks like, to be honest with you. I'll have to look that up. All right, now, don't worry about memorizing this. I just want to show you the different kinds of fruits and the way they're arranged. We've talked about drupes. They're called drupes if you have that big pit. Berries, and then you go, but wait a minute, on the right it says there's a berry. So you got to remember taxonomy and science. So anyway, legumes, those are the beans. Now, the important thing about the legumes, too, is that ecological thing you do. Remember, legumes are really good at fixing atmospheric nitrogen. As a matter of fact, we were just talking about cotton, right? cotton farmers in the old days, before this extensive use of fertilizers and such, cotton removes a lot of nutrients and nitrogen from the soil. Cotton is very demanding on the soil, right? So every once in a while, cotton farmers would grow legumes, which of course have these nodules, and they put nitrogen, they put fertilizer basically back into the soil. They don't use it all. So once you harvest the legumes, then you plant cotton after that. So you mix it in. The problem is economics, right? Cotton yields more money than legumes do, so you have to take a loss. The samaras, even though that looks like at the bottom, even though that could be confused with the gymnosperm seed, right? Remember the seeds that come out of pine cones kind of look like this, but remember, there is a fruit, there is a thickening around here, but in this particular case, again, like we said in the previous picture, it's probably not depending so much on the fruit to be dispersed. It's obviously depending on the wind, right? Aggregate fruits and multiple fruits. I would like you to look at those. This is, again, a familiar things that you eat, but sort of an interesting way of doing things. An aggregate fruit is like a berry, raspberries, blueberries, those kinds of things. If you look very closely at them, each of those little ball right there, okay, each of those little balls is a fruit. Each of those represents an ovary with a seed. Okay, so what you have here is you have a flower, okay, that has many ovaries. Now, I don't know if they have many, they probably do too. They probably have many anthers. Remember, in the pictures we've looked at so far, the drawings and all that sort of stuff, we've only been looking at one ovary here. See, that's just one. Remember, ovaries can be in whorls too, or the carpels, which is the ovary and the stigma and the style, the whole thing together. They can be in whorls. Often it's just one ovary, but a plant like the raspberries and the blueberries and all them guys, this is actually many ovaries in this particular flower. So you get one flower, lots of ovaries, lots of pollen, everybody's getting pollinated. You might make a bunch of little tiny fruits. Pineapples do it a little bit differently, though. In the case of the pineapple, the pineapple fruit is actually a bunch of flowers. Okay, so it's a bunch of flowers, and they form these fruits around sort of like a cooperative flower thing, right? The fruit that you look at in a pineapple is actually, like it says there on the right, the pericarp of individual flowers. Pericarp, the outer carpal lining, the ovary. So all those are ovaries, not in one flower, though, in many flowers. So that's the difference in the berries, right? The berries are one flower, lots of ovaries. This is many flowers. I don't know if each flower has a single ovary, not in pineapples. I'm not sure. Right, but the fruit actually fuses together. So if you think about it, cooperative flowers, right? This whole thing of cooperation, which always I seem to get really fascinated about. If you think that, in this case, you've got a big old fruit, man. Who's not going to want that? Who's not going to want to come and chew through that and drop seeds everywhere, right? So the more attractive you make yourself, like I said, for ingestion, of course, ingestion involves also sloppy eating, but it also involves, the whole passing through the digestive system. There's a lot of seeds, and we've talked about this, that have to go through the digestive system before they can even germinate. And of course, what we're thinking about is if you eat these things, as it's going through your digestive system, you're moving, right? So you're moving things around. Herbivores will store food together, so they will collect seeds from areas and put them into a herbivore cache. The old, I don't know, was it an ant, a grasshopper? That story that we learned about as kids, so you don't waste. I don't remember. One dude stored up food and the other one didn't. I always kind of like the guy who didn't store food. I figured that's more fun. I don't know. You can hook on things. Cooper, which that's kind of what Cooper looks like a little bit, but that's looks like a Springer Spaniel with barbs and hooks and those kind of things. And we talked about, did we talk about beggar ticks? Yeah, earlier, that had a little Velcro thing on there. You can attach yourself to stuff, right? That's important. So wind, you can float like the coconut seed that you see down there. However you make your way around, I'll go back to pollinators. However you get the pollinators to move around, flowers, nectar, etc. However you get the seeds to move around, blah, blah, floating, exploding, pooping, whatever you want to call it. However you do that, in the bottom, the result is what you see there in the red, right? You go lots of places. Move, 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 move. It's all about moving. It's all about making lots of things that move. Life is perpetual, man. It don't give up. It just keeps. It's like Arnold in the first Terminator movie. It won't stop. You can't stop it. Okay. All right. Now there's lots of unique things about these flowered fruited plants, but one of the things that's really unique about them is a unique thing they do. But it's really not so unique. You'll see what I mean in a minute. It's unique for plants. Let's start with the flower. I don't have my pen again, so I'm sorry. But anyway, start up here. The flower is a sporophyte. All right. So now we've moved way beyond big gametophytes. We have gametophytes, but remember gametophytes, which make gametes, are inside. They're all kept inside. And we'll get to that in just a second. So you got the mature flower. Let's go the male route. Let's take the anther. So, right, we're diploid here. Follow. The color behind it is orange, so that helps you remember where we are in the stage. Inside the anther, the male part, there's the micro word. We have microsporocytes. Those are diploid cells that through meiosis make, microsporocytes that produce microsporangium that through meiosis make microspores. I just got a little ahead of myself. Now, when you see the word meiosis, right? When we see the word meiosis, we know it's cell division. But the most important thing about meiosis is not that it makes more cells. It really, one process of meiosis, makes 4 cells, right? What's so special about meiosis? What do I always hammer when I get to this point? Those spores that are being made, there's something very, very important about them, and that's because of meiosis. What is that? Yeah. Right? And why are you doing that? Because it's all back to chapter 1. You're trying to make a bunch of different stuff because they're not all going to make it. But we'll follow the one that does, right? So we get a microspore. Now, you've seen spores before. Oh, that reminds. Sporophytes, like the flower. Sporophytes, like the little things that hang above the mosses. Guess what they do? They make spores. That's why they're called sporophytes. And when they make spores, they make them through meiosis. They make different spores, okay? And those spores divide and do their things. But what they eventually end up making is something we call a male gametophyte, which you're aware of. You've seen male gametophytes. Antheridia is a male, is found on the male gametophyte. Okay, there are male, the thing that looks like a soccer ball in the fern is the male gametophyte. The pollen cone in a pine tree, there's the male gametophyte. This is pollen again that we're making. It's in a pollen grain. Now, there is a tube nucleus, there's a tube cell, and there's something we call a generative cell. And we'll get to some of those in just a second. But again, don't get too hung up on all the names of those cells. It's really kind of more important to me that we talk about what those cells do. Anyway, the pollen makes its way to the stigma through whatever pollinator. And now you see why there's a tube nucleus tube cell. The tube nucleus and the tube cell form the tube that goes down the style. Okay, the style is a hole. It doesn't have to drill through, but somebody's got to follow through. So it goes down the hole, right? It goes down the style, blah, 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 blah. And this particular, there's lots of C's, but this particular one turns corner right here. You see, turns corner. And you notice that there are basically 2 nuclei, aren't there? Not counting the tube nucleus. The tube nucleus is gone. Actually, I think it's a little tiny blue if you look in the picture. But the 2 nuclei that we're looking at right now are these 2 right here. And I'll get to those in just a second. But I want to show you this turning the corner. So I'm going to come back to this slide, but let's look at it. All right. So on the left-hand side is the male part of a lily. We're going to look at these under the scope. So this will be good review. And there's the filament, the thing that holds it up. So what we've done is take the anther and cross-section, cut through it. Lily is an example of what we call a tetrad, all right, because there are 4 pollen-producing places, right? 4 microsporangia. I guess we would say microsporangia, right? Notice there are 4. They're pointing out to 4. Again, we'll look at this in lab. On the female end, we have, of course, the megasporangium, right? The thing that makes megaspores, and then they do their thing, and they form an embryo sac eventually. Now, the pollen tube in this particular picture has come down this route, and it it moved through something called the micropyle. I like that word. I think that's cool. What's the micropyle? The micropyle is the opening into the megasporangium. It's the opening so that the sperm can get in there. So, oops, wrong way. Yeah, no, right way. So each one of these little dumaflatches here has a micropyle that those sperm have to get into. Okay, and when they get into there, then something happens called double fertilization. Now, I'm going to come back to this slide, but I want to go and show you up close what happens in double fertilization. This is unique. It's, again, unique to these plants, or unique to plants, but not so unique when you think about it. So different picture, but I'm just following the cells, all right? So we got an ovule, we got an ovary, and what we're doing is we're first going to make the egg cell, right? I know we've been following the pollen, but you can't follow what the pollen does until you follow what the eggs have done. So I'm actually very quickly moved through this part. Okay, so the megasporangium divides, and when you undergo meiosis, you make 4 megaspores. All of them die but one. Now, this is what that one cell does. So I'm describing what happens when you get this. All right, so that's where we are. So kind of remember that picture. Looks similar right there. All right, so here we are inside of the megagametophyte, right? The female producing thing. All right, now, so that one surviving cell undergoes mitosis 3 times. Now, mitosis, when it undergoes 3 times, you get 2, 4, 8. So remember that word, 8 cells. Let's account for them. All right, so you get an egg cell. That's the one in blue. There's one. You get cells that occur on either side. See the 2 little dots? So I'm going to move between the thing and the yellow down below so we keep track of those 8 cells. You got 1 egg. Those 2 that are on either side are called synergids. These things right here. That's a synergid. That's a synergid. Now, it's a cell, but it doesn't get involved in, it doesn't get fertilized. All right. The reason why it's there is it actually guides that pollen. See, there's the micropyle. It actually guides that pollen tube through that little hole that we showed you in the picture, and then it says stop. It says you're here. Stop. It's kind of like when you know, your GPS says arriving at location. You're like, where am I? Right? So it says stop. You're at the spot. Release the sperm, please. So those 2 release the sperm. So the 2 sperm come out. Now we'll go back to the sperm in a second. So we got 2 synergids. Got an egg cell where you got 3 so far, right? So as we move up, we get 2 more cells. Now they call them polar nuclei, but they are cells. All right. Now, those polar nuclei are the important part, the really interesting part, the part that's unique to flowers, the part that's not unique to life, although which is what I find interesting. You'll see in just a minute what they do, but they form something called the endosperm. Now, I did talk about endosperm before. Do you all remember what endosperm is? I said that in flowering plants, I showed you a piece of corn that had been cut open, a kernel. Do you remember what we said endosperm is in the seed? Yeah, it's inside something, but you remember what, like in popcorn, I said when you popped popcorn, that was the white stuff that fluffed out. Remember, that's the food, right? The endosperm is food. It's food for the seed. Now, we can take advantage of it. When you eat the inside of a nut, you're eating the endosperm. That good-tasting walnut is really intended for the seeds that are in there. All right, so we'll return to those in just a second. Again, that's maternal care is what I'm trying to get at there, right? That's working on producing a lot, don't get me wrong, but fewer young with more care. Because even though plants produce a lot of seeds, a lot of pollen, you know, all that sort of stuff, you see it if you're allergic, they don't produce the quantity that you got to make if you're a moss, right? Because you're really living on the edge of the ability. Well, maybe moss people would disagree with that, but you're living kind of in a very special environment. We're talking about organisms that can survive anywhere. All right, so how many? 2, 4, 5. We need 3 more. The antipodal cells, these are easy, so we can stop. And we don't know what they do. Not quite know. I read some stuff that has something to do with on the left, you see a picture of the thing I'm talking about. Some of the, there's the synergids at the bottom. Some of them are labeled. Anyway, those other ones I've heard might have something to do with forming these chalazoid cells, which I think is something helping orientate the embryo. Anyway, don't worry about it. We don't exactly know what these 3 at the top do. We know what the synergids do. We know what the egg cell does. Obviously, that thing gets fertilized. Let's go back to what those polar nuclei. So let's go back and talk about what these guys do. During the process of fertilization in an angiosperm, we call it double, not single, double. The reason why it's double, I'm sorry if I'm going to flip back and forth, is remember there were 2 sperm that were traveling down there. There was the tube cell and tube nucleus, which made the tube. Then there were the 2 sperm. So here come the 2 sperm. The synergid said, release those 2 sperm. 1 sperm fertilizes the egg. We'll get that out of the way right away. So you get your diploid offspring. So you get fertilization. Boom, boom. Just like in everything else, right? Now, here's where they're different. You got double fertilization. The other sperm doesn't fertilize really, but technically what it does is it combines with those 2 polar nuclei. All right, so what it really makes is it makes an endosperm that has a sort of a unique chromosome number, right? It's not N or 2N, it's actually 3N. 2 polar nuclei bring in their end, and then of course the sperm that fertilizes. So that's what the other sperm does. Now, the reason why this is important or crucial or really unique, if you will, is because when that happens, you have formed a starter sac for that seed. The bigger and more complex your endosperm, so now I'm falling back into the diploid, the bigger that endosperm, the more nutrients that is, the better chance that junior zygote is going to make it. And that is significant, folks, right? That is like the ultimate in dispersal. I'm going to make things that attract insects, or maybe use wind. Then I'm going to make things, maybe I don't. Maybe I'm like the maples and make the little tiny fruit, but I'm going to attract things that make things want to eat me and move me around and drop my seeds. And then when those seeds happen, I'm going to give them a chance to survive. I'm going to give them a starter pack. Do all seeds make it? Well, no, you know they don't, right? Lots do. Lots more seeds than spores. Lots more seeds. Oh, there you go, moss, in your face. Lots more seeds survive than, say, the spores of a fern up there. Look at the sori. I mean, if you count it up, you know that you make more sori than you do seeds and pollen and all that. Maybe not pollen. I think maybe I told my other class. Male gametes are cheap. That's why we make lots of sperm. Females make the important gamete. All right, so what do we get in a seed? Well, we get a seed coat, and it's diploid, and it's tough, and it's strong, and it's durable. Oh, that's just like a spore, only better, right? We get the food mass, endosperm, the popcorn that we eat. We get the developing embryo. We get the junior who gets to feed off the food mass. And if we make 1 or 2 leaves, we're 1 or 2 of these. If you make 1 leaf, a cotyledon is the cotyledon. So what did he just say? Is what we call an embryonic leaf. You either start off with 1 leaf or 2 different plants. We'll talk more about these. They're called monocots and dicots. Where am I in time? Oh, good. All right. Any questions about that? So the big things that are going on here is going to be this this double fertilization, right? That's we haven't seen that yet. We've seen seeds, right, when we looked at pine, but we haven't seen double fertilization. So while pines kind of, help themselves out by being able to disperse and producing these seeds. Remember, this is the first time we saw seeds was in the pines. So while they helped themselves out by producing this resistant structure, these guys took it to the next level, right? They said, we'll take your resistant structure and up it one. We'll not only make a resistant structure, but we're going to give the embryo a little food pack, kind of like your lunch pail when you went to school, to give it a start. So that when it gets out there and tries to germinate, it doesn't have to immediately find resources. It's got a little bit to go. So it can maybe wait. Maybe I got released. Here I am on the seed. I got released today. Oh, man, too bad it's not. There's no water. Oh, it's going to rain tomorrow. Maybe I can last till then. Maybe I can. Maybe I can live where it's drier. All right, we'll look more at that when we get into the lab. We talked about this. Where did these things come from, these flowering plants? Poor Darwin. Poor Darwin. He said that up above, too. Remember what he said about the Cambrian explosion? He said the same thing about what we call the angiosperm explosion, I guess, about 100 million years. He says it rapid, relatively rapid. So he looks in the fossil record, and he found all those animals almost instantaneously in the Cambrian explosion. You know why now. Remember, evolution isn't just gradual. It is, but sometimes it's pretty quick. And then in 100 million years, here come the flowers. He digs up and goes, oh my gosh, it happened again. And again, he knew gradualism, but he didn't realize that there was another pace, right? There was another pace you could engage in cladogenesis. Remember, where like species, is changing maybe a little bit, punctuated equilibrium. Boom, and all of a sudden, a bunch of species. All of a sudden, relatively speaking. All right, so when did they start? Well, 125 million years ago. So we're still in the dinosaurs, but actually that's probably not the very first beginning. It might have been even before that. These things didn't fossilize really well. It's hard to know when, there were things out there that kind of maybe looked like this. It's the fossil record, and we're not dealing with hard bony structures like on animals. These fossils that you see, Arctofructus, 125 million years ago, Monsecchia, more than 125 million years ago. That's what we think Arctofructus looked like there on the right. We thought it had like these little thin leaves, stamen, right? So that's the male part, carpal, what we thought may have been floats. Now it doesn't label on here what would have been the fruits, but and Monsecchia down below, this one down below, the one that was even older, we think these might have been the fruits. The floats on this guy is kind of cool because it tells us something about where it might have lived. We think maybe it lived like this, like in shallow water. These aren't pineapples, but maybe the early evolution of flowers or something like that. So these are the oldest known angiosperms, or oldest known. So where Darwin saw them in 100 million years and kind of went, wow, all this really cool stuff happened in 100 million years. That's kind of fast. He didn't realize that all this stuff had been going on in other ways. Remember, it's not ladders, Aristotle, right? Things blend. Things change, sometimes fast, sometimes quick. All right, now, where are we today? The ones in the dotted line are extinct. I'm sorry, not extinct. The what do they call them? Venetales. So I'm not going to talk about those, but I'm going to talk about the one in red, Ambarella. Ambarella is really cool. For one thing, Ambarella is kind of like, it's a flower, it's a plant living fossil. Ambarella, if you look at it, is a basal taxon to that most recent common ancestor, right? If you start right there where there's the blue, ignoring all this, if you start, remember, you decide where the group starts. So if you start right there with the blue, right there, we say that this is a basal taxon. Remember, it's a taxon that's around today that comes off the main line. Right? Basal taxon. So it presumably looks most like what the ancestor looked like. Now, it's been a lot of time, 150 million years, I get that. But it's nice for one thing that it's only found in these rather limited locations. In the lower right that's Australia. So this would be, what is that, Caledonia, the Caledonian Islands. I think where Guadalcanal is, some war that was fought in World War II. But regardless, Ambarella is only found in that area of the world. And Australia has weird things. Australia separated from the rest of the world a long time ago. So the weird things that were around back then, many of them, in one way or another, kind of survived. So that's why we think Ambarella might be. And there's Ambarella. Now, not a very pretty flower. Can you imagine if you were, somebody brought you flowers, right? Or if you were buying flowers for somebody. Here you go. Here's a bouquet of Ambarella. I wonder if they even smell. It looks like it's like a flower that's been chewed on. So anyway, it's not pretty, but we do think that that's maybe what the first kind of flowers looked like. And remember now, these are modified leaves. So again, it may not look exactly what something we would see today. All right, so once we took off from there, then we move into other things, water lilies, the star anise, and their relatives. A big group of things we call magnolids, which, believe it or not, as you can imagine, the magnolia love magnolias. And then what we call monocots, and I call them dicots. They're not called eudicots. We'll talk about that in just a second. So I'm going to move in now and talk about these different groups. We've already talked about Amberella. I'm moving to the water lilies, but I'm actually going to do it in reverse. I just looked at my next slide and decided that I should do this first, actually, so when you get confused. Let's talk about the difference between a monocot and a dicot, or a eudicot. If you're thinking there's something with a number, you're right. And if you're thinking cot, I've seen that word, you're right, because a cotyledon is a primitive leaf, right? I just wrote it, embryonic leaf. So these are the plants that when they are back in the seed. Right there, here. If you start off with one or two, you're one or two of these. Cotyledons, you have one cotyledon, you have two cotyledons. All right, so the monocots and the dicots. Let's look at the difference between them. Nice little chart side by side. I've got one in lab too. Used to be called the monocots and the dicots, but the dicots actually ended up being a paraphyletic group, so it was a false name. It's like protozoans. So now we got what they've redone things now. Now we've got the eudicots and the monocots, and in spite of all these characteristics, you think, my gosh, remember all these to remember apart. It's really simpler than that. The eudicots are bigger. There's about 170,000 species, and these are trees and shrubs, beans, flowers, things we eat, trees, stuff like that, right? Monocots are like rice, wheat, grasses. Wheat is a grass, really. Bananas, palms. If you think about it, even though bananas and palms get real big, isn't that kind of funny? Palms are not true trees. Dicots are trees. Palms are big, thick blades of grass is really what they are. I have a cross-section of a palm in class. I'll show you. It doesn't have the rings like on a regular tree. It looks like a bunch of grass been mashed together. Okay, now these characteristics. What am I time? I'm going to go over these in lab. So you go over them in the meantime. But again, probably for me, the best way to remember a monocot from a dicot, I do want to show this because I know you guys are working on your field notebooks. Dicots usually make big flowers because they use pollinators. Monocots, like remember I said your grass, your St. Augustine has flowers. It might be small. Use small insects or wind very often. Easiest way for me to remember it, it talks about the number of floral organs, number of petals, number of stamen, number of carpels, whether it's in 3s, right, or 4s and 5s. You think that'd be the easiest one, but these things fall off sometimes. You see a flower in the wild, and it's a monocot or a dicot, and you count the petals, and it'll be wrong because one of the petals is missing. So you got to know what you're really doing. I look at the leaves. To me, that's usually the best sign. If you look at the leaves up to the light in a monocot, the leaves are straight and parallel, and if you look at them in a dicot, they're like a maple leaf, right? They're netted venation. All right, so we'll go over that again in lab, but that's probably the best way if you're telling them now. Don't rely so much on the numbers. I would definitely start with the leaves. Can't probably see the cotyledon unless you have a microscope on hand. All right. There we go, basal. So let's look at these guys. Basal angiosperm. So the Amberella is a basal taxon. Water lily, star anise, those are all considered basal taxons. Amberella is the most basal, okay? But what we mean by that, the significance of using basal to describe these, there's only 100 of them, 100 species, is the fact that we think these look a lot like the original flowers, okay? They look a lot like the red. So Amberella is the one that looked the most. Water lilies, that's the water lily in the upper right. You see those in the pond on campus. Those are common Florida water lilies. American, I see these all the time in people's field notebooks. Star anise, I don't know. We have anise relatives in Florida, but I don't know if we have the star anise. But it's a very distinctive looking flower, obviously. And flowers are oftentimes very colorful, too. All right, how about the magnolids? Okay, the magnolids are a huge group, about 8,000 species. It includes the magnolia tree with beautiful, I have one in my backyard. They don't grow very fast, unfortunately, but man, they make a lot of really cool flowers. Beautiful smell. Look at I love the name too, Magnolia grandiflora. Isn't that neat? Big flower. There's this little girl that used to live next door to us, Ava. She was just learning how to speak, and she could speak really well. It was amazing to me. It always used to crack me up, the stuff she said. So one day I brought it over to one of the flowers, and I said, what's it smell like? I figured she'd say, oh, good or sweet. She said, like lavender. I'm like, how many kids our age know the word lavender and what it smells like? That was pretty impressive. Pepper, that's what I'm showing you up right. That's how pepper grows. Pepper is a magnolia. Oranges, Persea americana is the orange. Obviously, these aren't ripe, but those are the magnolias, about 8,000 species of those guys. And that's not the biggest group. All right, so we got the monocots. That's a pretty good sized group too. Monocots, now we go from 8,000 species, or about 8,000, to 70,000, or about a quarter of them. And these are the grasses, which can make flowers, and sometimes pretty good sized flowers. Like orchids belong in this group. An orchid is a grass, it doesn't grow as a tree. Orchids are often considered epiphytes. They grow on something else. They grow on another tree. Doesn't mean they're a parasite, by the way. They would be more of a +0 or commensalism, right? I put that little note in there because I went out in the woods, Lake Louise, at one time on a field trip, and there was these beautiful orchids growing in the tree, and I was able to show the students it was a native Florida orchid, right? It's gorgeous. And then we came back the next time, and somebody had removed it. Somebody had picked it. Somebody had taken it, which was kind of awful. Orchids are protected, folks. You can get in trouble for picking orchids. But again, these are the things like the palms, all right? The palms in the upper right, the big tree one, that's the Florida state tree. That's the cabbage palm. And if you look at the leaves on the cabbage palm, or the fronds, if you will, they have sort of a V shape to them. Or keeled is the word we say. And then the other one is the saw palmetto on the right. Both of these are native. A lot of the other palms aren't. A lot of the other palms are Middle Eastern palms. But the saw palmetto, the leaves don't have that sort of V shape to them. Okay? All right, grass belongs in this group, 70,000 species. But that's not the biggest group. Biggest group are these guys. These guys are the dicots, okay? The dicots are 170,000 species. So this is the big group, okay? This is the big roses, swamp rose, which is in Florida. Beautiful. Oh, you can now remember why I did this picture. All right, swamp rose. Here, horrible name, beautiful flower, right? Rosa palustris, much better. The whorl of the petals, right? There's a whorl. The whorl of the stamen. The whorl of the carpels. I can't see the sepals, but that'd be in a whorl too, wouldn't it? All right, very lovely flower. You can actually even count this one too. How many foot? 1, 2, 3, 4, 5, right? And if I bet you counted, unless one of them had fallen off, the carpels and the stamen, you would get a multiple of 5 as well. All right, Florida Alicia is another flower belongs in this group. Chapmania floridana. Now, something very significant about the Florida part being used so much, not only in the common name. These grow on campus. You'll see why that's so cool in just a second. I've seen them growing on campus, and the Florida in the species name is because this particular flower is endemic to Florida. What's the word endemic mean? Why is that? Why am I making such a big deal out of being endemic? You might say polar bears, okay, right? Native, give me a little more. Some things we have in Florida are native in Florida, like they've been here all along, but they're also found in Mississippi, or they're also found in the next county. Sometimes we find organisms that are found right here. In a really big way, we could say polar bears are this way. Yeah, right? Polar bears are endemic to the Arctic. Now, I know the Arctic is a big area, but it's what you say. Florida Alicia is endemic to Florida. You know what that means? It's only found in this state. Now, I can't remember in the picture if the light yellow is where they're higher found or lower found, but it's not found outside the borders of the state of Florida. That means from a conservation point of view, this species has a very high probability of going extinct, right? All you got to do now, luckily this is a relatively hardy plant, but all you got to do is change a little bit about its habitat, and not only do you kill off, I don't know, 10, 15,000 Florida Alicia, but they might be the only ones on Earth. Now, why is that bad? Because it removes the diversity on Earth. And we live in a world in which diversity is important. I spent 2.5 months talking about it, right? So we're removing diversity. And the more endemic something is, something might be endemic to you. Well, there isn't, but hypothetically, to your backyard. It's only found there, right? The eudicots, really big group. Not all of them are really big, though. So we got to think about that when we do it. Now, the number one thing that causes species to go extinct or species to go in peril, the number one thing, you say, well, they're dying off. Yeah, I know. People shooting them. People eating them. Nah, it's habitat. Habitat destruction. I'm typing it. Habitat destruction is the number one reason why organisms go extinct in the world. You change their habitat. Now, sometimes the habitat changes on its own, like the dinosaurs. What we were around, ain't us. Don't blame us for that, okay? But when we go in and we do things like pave, and we add fertilizers, and we change waterways, and we do things that we're not sure about how it affects the organisms, we sometimes go, oh, crap, we just killed every one of them. Now, does that mean the world's going to end if all the Florilegia are dead? No. It sucks. Sorry. I mean, some botanists could argue, well, now the soil's going to become messed up this way. That's fine. And I agree with that and support that. But to me, the different the better. Legumes, we eat a lot of those. Those are important. Those are eudicots. We eat a lot of this stuff, right? 80% of the calories we eat. 80% of the calories we eat as humans come from these kinds of things. Wheat, rice, maize, potatoes. So seed plants are pretty doggone important, aren't they? You knew that. Did you know that they're really, really bad compared to beef, though? Really, really bad in the terms of beef is a waste of these organisms. Beef is a waste of seed plants. 5 to 7 kilograms of grain, grass, hay, right? Those kinds of things by the cow gets converted into 1 kilogram of beef. 1 kilogram of beef. Okay, so what? Let's say a human being could eat a kilogram of grain. What 5 to 7 people can eat is now can only feed one person. Now, yes, I know the caloric contents are different and all that sort of stuff. So don't focus so much on the numbers other than the idea that eating beef is very inefficient. It's much better to eat the grain. Now, I don't eat beef. I've stopped eating beef a number of years ago, but I eat steaks and I like them, okay? So if you eat steaks and you like them, I get it. I understand all that. I've always preferred fish and all that. That's the only meat I don't eat is well, dogs and cats either. But anyway, what we're saying here in the developing world, in which these people are really trying to maximize what they're getting out of their land, right? The best thing for them to do is to grow agricultural products on it. What goes down in the Amazon rainforest is they clear the land because they grow cows. Cows bring more money, so they grow cows, or they allow cows to wander around. The cows destroy the habitat. They nibble the roots down to crap. And not much grows in the Amazon rainforest because the soil is actually kind of crappy. It's hard to believe with all that stuff growing down there that it's crappy soil. You would think, oh my God, it must be rich. It must be that black, rich stuff. Most of the nutrients that makes that soil black and rich is not in the soil. It's in the organisms. It's in the trees because, man, things grow. Things decompose and get back into the living really fast down there. So the soils, they don't hold a lot of this stuff. So when the cows don't have anything to eat, guess what the farmers do? They cut down more land. All right. That's my soapbox kind of thing. We also get some good medicines. Actually, no, I'm not done with that. I got another whole thing to talk about that. We get atropine, digitalin, methanol, quinine, good for malaria. Taxol, ovarian cancer drug, right? Curare, tubocurarine, it's a muscle relaxer. The Indians down there used to use the tubocurarine from the curare tree and put it on their blow darts and then shoot things with it, like small mammals. And they could feed upon them. World's largest rodent lives in South America. The capybara. You ever seen a capybara? At a zoo, maybe? They're a neat critter. I'll show you when we get into the animals, but they're real common at the zoo. They do real well in zoos. Look like huge rats. They are. The world's biggest rat, world's biggest rodent. But man, you could probably eat a lot off of that. I should have just waited to use up my energy on this. But what I'm trying to tell you with all of this is each year down in the tropical rainforest, there's an example from Brazil. You see the time span. 55,000 square kilometers are cleared every year. So the green, the nice lush green on the left is forest, and then all the stuff on the right that's not green is going to be humans, okay? 55,000 square kilometers. What does that mean? West Virginia. Every year, the size of West Virginia, or area rainforest that size, gets diminished, right? Losing their habitat. We're losing half of the species down in the tropical rainforest are going to be extinct within the next few centuries. Does that matter? if we keep doing this at the present rate. Does that matter that half of the species will go extinct down in the tropical rainforests? It's not a rhetorical question, Marie. I mean, it's going to be a lot of insects and little tiny fish and all, but who cares? Of course it matters, right? Why does it matter? Give me just one negative aspect of losing these species, or losing one of these species. You're going into healthcare, right? Physical therapy, was that what it was? Yeah. So sometimes in physical therapy, if somebody's dealing with a lot of pain, you might prescribe a medicine, right? And one of the big problems with medicines, of course, my dad has MS, and I know that with him, this is a situation, all the doggone side effects that occur. What we find with a lot of these things that live down the tropical rainforest, for one thing, the Indians have been using them, of natives, for many, many years. And then these things take care of the problem without the side effects, or with minimal side effects. Not only that, there's the idea of a keystone species. Now, we'll talk about more of this in animals, but the keystone species comes from the idea that when the ancient folks used to build arches, right, it was oh, God, Steve, you and your artistic ability. There was that one stone at the top, right? Arch wasn't glued together. You pulled that stone out. That stone kept the arch together. That's the keystone. In ecosystems, there are sometimes keystone species, or species, more than one, which means that if that species is removed, or that species increases in number, or if that one species does something different, it affects the whole ecosystem. Now, we go down the tropical rainforest and we kill some little bug. It's the butterfly effect, that movie with, Kutcher, Ashton, something or other, right? Butterfly effect. You go back and you kill that one little insect, and that one little insect fed this bird, and the one little bird was used by these organisms, and and you trace it all back. Before you know it, you got a big ecosystem collapse. Okay, we only got about 200 years of these things left. We keep cutting them down, and like I said, we keep cutting them down because the soil sucks and we're feeding cows on them. And one of the biggest problems, of course, is this fact that big trees do what they do in photosynthesis, right? You talked about that in Bio 1, right? They take up carbon dioxide. And guess what happens with that excess carbon dioxide that's not getting taken up? Drifts up into the atmosphere, and it forms a blanket. Now, I read the other day online that there's this group of folks out there that are arguing against climate change and saying that rising carbon dioxide has not been proven to cause the heat of the Earth to change. All right. So remember now, it used to be called global warming, but global climate change is a better way of looking at it because it's a global disturbance that's occurring. Part of it is the warming in certain areas. It may be nice and warm down here, or it might be nice and cool in the Midwest, you know. So where's this global warming? But it's still cold, but a little bit too warm in the Arctic. So the Arctic ice caps start to melt, right? You get the idea. Global climate change is going on. So the argument was that the temperature doesn't cause, carbon dioxide doesn't cause the temperature to do that. And that's such crap. Venus. The reason why Venus was a planet that was very similar to ours in terms of being able to support life, a little too close to the sun. But the biggest problem with Venus is it's got this thick layer of carbon dioxide around it, which traps the heat. Venus is hotter than hell. Liquid flows like water on Venus. So doggone right it does. Of course it does. We have evidence to show it. In the last number of years, the carbon dioxide in our atmosphere has done something like this, called hockey stick. Blip, blip, gone up like that. And guess what? When you go back in that time, guess what happened at that time? The Industrial Revolution. Industrial Revolution, right? Was when they started using factories, burning coal, dumping fossil fuels. Come on, folks. 97% of all scientists who study global climate change confirm that anthropogenic characters, this is the exact thing they're saying, characters that anthropogenic factors, things we do, burning fossil fuels, cutting down forests, things we do, 97% of scientists said that is contributing to global climate change. Contributing. The Earth changes and goes through climatic cycles. We're saying what we're doing is making it worse, making it happen faster, making it happen at more intensity, all right? And making it happen in such a way that it can't recover. Because the cycle up and cycle down, we tip the point, and then it doesn't recover, and we've got a global catastrophe on our hands. 97%. And yet politicians stand up and make comments about, well, I'm not a climatologist, but I don't really believe this global climate stuff. I tell students, if you had a heart condition, we're going to die of a heart attack, how many cardiologists would you need to go to before you believed it? Maybe 2, right? Second opinion. Would you go to 7, 8? Would you tell the 12th cardiologist that says you're going to die of a heart attack? All 12 of them said that. Let's say 11 have said that. Would you tell the 13th one? Would you tell them, well, I don't know. I went to 11 of y'all, but I still don't believe I'm going to have a heart attack, all right? I'm not going to talk anything about this slide because this is a learning aid. This is a slide I put together when I was learning these. I know you're thinking, whoa, this is really fancy. It's not. But it was a way that when I was going through the plants back when I was much younger than I am now, I made a little graph like this on a piece of paper because there weren't computers around, not for us to use. And I wrote out the features and I wrote out the different organisms. The only thing I would change about this now is I would change this word from conifer to gymnosperm. I'd change this to angiosperm. I'd use the right words for it. Moss and fern can stay, right? So use this as a way to go through. I've talked about everything on this. Use this as a way to go through when you're studying the differences between these organisms. Really good question for, one of my favorite questions, right? What's one of my favorite questions? What's one of my favorite type questions? That's it for chapter 30, by the way. If you have questions on that, you can give me that. You got it. That'd be good for a question like that. Okay, so 31, that's the fungi. Now, 31 is a weird chapter. Let me make sure I did that with you guys. Because at the end, the concentration of information really starts to rise. What I do with you all is 31 is not on any exam. Your next exam is 29 and 30, the plants, and then it's the animals. So you don't get a lecture exam on 31, or you don't get questions on it, but it is on the lab practical. We do a lab on it. And the stuff I ask about is on the lab practical. So, so this lecture is important to listen at least before the next lab practical, okay? Okay, so what I finished in the last chapter is the second chapter for your next exam. You only have 2 chapters on the next exam. It's only on plants. That's a lot, but it's at least on one subject. Oh, man, I love fungi. These grow in Florida, too, by the way. This is Lactarius indigo. I love fungi because they're colors. They're a pain in the butt to identify. They're horrible to try to identify because they can take on any shape they want. But, man, I tell you, sometimes I saw one of these out in the woods behind school one time. And they don't last very long. That's the unfortunate part about them to show you. But it had this beautiful color. And it was like when you broke it up, it had like this thick substance in it. A little thing on the color here, these are spores. Fungi are these spore masters, man. We've been talking about spores in plants, and these guys, they're really good with using spores. This is that stuff that grows on, doesn't grow on trees. Well, this one does. This grows on, this is what we call a bracket fungus. It looks like a bracket. It looks like, a thing that holds up a shelf, right? This particular one has a really cool name. It's called turkey tail. You've seen them. Maybe you've got it in your field notebook. It looks, look at the colors. It looks like a turkey tail. There's lots of things that grow as bracket fungus that aren't turkey tails, okay? All right, let me get my other slides here going. 31. What's the weird thing about these critters? Oh, there's too much to just do in the time I got left. We're going to spend a lot of weird things. For one thing, one of the weirdest, interesting thing, the largest single organism in the world, the largest single organism in the world, the largest whale, the largest tree, the largest blah, blah, the largest single organism in the world is a mushroom. It's a honey mushroom which grows in Florida. It's not something rare. It's not something rare. Armillaria oysterii, they're called honey or oyster mushrooms. I see them growing in the woods. But this particular one, this one oyster mushroom, covers 9 million square meters of forest, close to 2,000 years old, weighs hundreds of tons, 9 million square meters, something like 3 miles, I believe. And if you're thinking, oh my God, that's the biggest damn toadstool I've ever seen in my life. After you learn about fungi, you're going to understand what I mean by how an animal can, or an organism can be that big and not be a 3 mile across toadstool. See, the thing about fungi is they live a really bizarre underground life. Most of the critter is underground, right? So more on that later. 1.5 million species. We have single-celled varieties like yeast, penicillin. No, penicillin is not single-celled. Yeast is single-celled. We have multi-celled varieties like penicillin and the mushroom you see there. We have those that reproduce sexually, more typically, especially in some species, asexual reproduction. All right. Number 2 weird thing about them. Number 1, the biggest one in the world is one of them. Number 2, they digest food on the outside. okay? We do it on the inside, in our stomachs, animals. These organisms digest it on the outside. What they do is they secrete exozymes, so kind of like they secrete this material with enzymes that digest the food in the environment, and then it becomes digestible. It's like already been chewed on the outside. Now, this is important for ecosystems because guess what? These exozymes make a lot more digestible material than the mushroom itself, even if you are 9 million square meters, can use. So they enrich the soil. So more on that later on. Of all the critters out there, animals, plants, all that sort of stuff, animals and plants anyway, not of all the critters, we're mostly closely related to these guys. We're more closely related to a fungus than we are to an animal, or to a plant, to an animal, I'm sorry. We diverged about 460 to 670 million years ago. Many folks I just don't think would think that. You know, they make a pretty good group of all the critters on the planet too. See that pie chart? Many folks would think we're more closely related to a plant, but it isn't. It's the weird fungi that we're closely related to. All right, so the exozymes I talked about, the exozymes can, like this bracket fungus, isn't that pretty? Can break this stuff down. This is lignin. Lignin, oh, there are some bacteria that can break down lignin. Lignin is the stuff that makes wood hard, right? It makes organisms be able to stand up against the force. And I always say that because I think about that movie again, about that guy, Trees Made of Wood. So this is that really durable organic material that only few critters can break down. And man, if you look in, there's lots of carbon atoms. Carbon atoms are important for living things. That forms the bricks to build the structure. So without lots of carbon atoms, so this is a lot of carbon atoms. You can't use them in lignin. They got to be broken up, and these critters can do it. Good for ecosystems. Mycelium. What is the mycelium? Well, the mycelium is the key to their survival. Okay, what fungi do, the part like I'm talking about that you can't see, the underground part, this part hidden in the branches of the tree, is they form this network of cells. All right, now I'll talk a little more about that later. But this network of cells does a lot of absorbing. It's got a lot of high surface area, so a lot of absorption. So these guys break down the lignin, they break down cellulose. Us animals can't do that. Some fungi are even carnivorous. That's kind of cool. Look at that in just a second. Some fungi actually eat animals. All right. So, all right. Now, 1, 2 weird things about fungi, right? Exozymes, big. Number 3, and the weirdest. In fungus, it's not about the cell. Fungus don't have cells in the traditional sense of cell. All right. Fungi are all about the nucleus. Remember that the cell is just something that keeps the nucleus combined. In fungi, the way they grow and the way they've made their life and the way they evolve is they don't worry about a thing that has a nucleus. They keep the nucleus because it's got the genetic material. They have all the stuff that supports the nucleus and keeps it alive, the ribosomes and you know, all that, mitochondria and all that sort of stuff. But they don't have cell membranes, okay, in most of them. Some of them do. What they do, though, is they very often, these little things that grow on the ground, this mycelial mat, forms these things called hypha. Hypha is a singular of a mycelial mat. Now I'm going to go back and forth between 2 slides. So in this picture down here, these are a bunch of hypha. And the hyphae grow and grow all on the ground, notice, and they grow. And the white that you see scattered among the debris and the dirt, when you pick that blue fuzz off of your hamburger bun, that is the hyphae. That is this mass of hyphae, this mycelial mat that runs out in the environment and guess what? Secretes exozymes. Okay, secretes exozymes and then sucks that stuff back up. So, wow, man, lots of surface area, right? Huge amounts. I got to even, is that the next one? Yeah, look at this. Look in the background. Look at all those hyphae spreading out. Lots of surface area. And when they're really small, you can see they really maximize that surface area to volume ratio. Lots of absorption going on. Lots of ways or lots of ability to move materials out, the exozymes, and suck the stuff back in. Now, without a cell wall, without a cell membrane, you can grow really fast. All right. So, all right, back to synocytic and septate for just a second. Some of the hyphae are septate. You can see it under a microscope. Some are synocytic. I know you want to say conocytic, but it's synocytic. Synocytic hyphae have a cell wall and the nuclei are all mixed together. Septate hyphae have septum with a pore. It's not a cell membrane or a cell wall. They only have that outside cell wall, made of chitin, by the way, kind of interesting. But they have a hole that things you see in the lower right can go through. But this is still basically one cell, and that's how many fungi do that when they're multicellular. So they form these hyphae of a cell, or sometimes in some species more than one cell. And in some cases, they're open like the Cenozoic, or they're closed like the Septate. Now, growing really fast. When conditions are right, what these things will do is they could just pump them full of liquid, and they're going to grow like the pseudopods on an amoeba. And they can grow up really, really, really fast. They grow really, really fast underground, and then when they find it's right, they can form a fruiting structure, a spore-producing structure, a toadstool. See, so all those hyphae are underground. So that big mushroom that I talked about that's, what did I say, 3 miles across? It's not a big toadstool, but the mycelial mat of that one individual, they use DNA to figure that out, spreads out 3 miles. And when conditions are right, these mushrooms pop up to the surface. When they say, oh, it's time to reproduce, they pop up, and the thing that pops up, the toadstool thing we eat, is the reproductive part. So the hyphae in the mycelial mat does the food digestion when it's time to reproduce. And look what the toadstool's made of. Look what that mushroom's made of. Hyphae. When you rip them apart, they're fibrous, aren't they, right? If you ever ripped apart a mushroom, we will in lab, and they're fibrous, and those are all hyphae that are stuck together. Lots of surface area. Now, we'll get back to the reproductive structure here in just a minute. That's important. It's different in different kinds of fungus and what goes on there. Well, maybe not just a minute. We only got 5 minutes. But anyway, we'll get to it, obviously. But it's the hyphae that I want to still sticking with, right? Hyphae are the key to these organisms. There's some cool pictures down below of Cenozoic and Septate. Pretty obvious, aren't they? Especially when they're stained. But the whole idea of being Cenozoic, or even Septate with the pores, because you can let things through pores, is they can very quickly flood those hyphae with turgor pressure, with water, liquid, and cause them to bulge up. And when they bulge up big enough, they can bulge up and form a toadstool. That's what a toadstool is. A toadstool is an inflated bunch of hyphae. All right? Now, they could do that fast. That's why sometimes it rains. You know, at night, I bet you if you go up my yard today, since it rained last night, you're going to find mushrooms growing. You're like, wow, these came up pretty quick. That's why. They're ready, man. They're going to make the most of this. It's rained. It's ready to reproduce. Okay, I talked about that. Now, the cell walls of hyphae, chitin. That's kind of interesting. That's a bit, just a little bit, but that's a bit of evidence that connects us animals to fungi and not plants. You don't find chitin in plants. You find chitin in animals, okay? Arthropods like that rhinoceros beetle, its exoskeleton, that's made of chitin. All right, you find that in animals. You don't find it in us. We can't even digest it, but you find it in animals. So that's, again, that detective work part. Wow, chitin's a very specialized substance. And it could be a convergent evolution, but other data showed just along with the chitin that it was the animals that were more animals and fungi more closely related. You just don't go on one piece; you go on many. All right, parasitic hyphae. Check this out. They're a little nematode, little worm. So we're talking at, you know, a quarter of a micron. So this is, you don't have to worry about walking your dog through the woods and a bunch of mushrooms attack. Can you imagine? A bunch of little toadstools running after you. But the hyphae, since they can grow really fast, can wrap around these little tiny worms. It's like a lasso. So once they've wrapped around these little tiny worms, they just pull them in. They get the worms sitting up on this bed of hyphae. They release those digestive enzymes, and basically the nematode dies a very slow death. Probably. Doesn't matter, nematodes. There ain't much to them. They don't have a brain. Leucin grows really fast, less than a second, so don't get close to one of these if you're a nematode. They can also parasitize plants, and what they do by that is the fungal hyphae actually grows and forms this thing called a haustorium. Plural is the other word, haustoria. And the haustorium spreads out spreads out, it pushes, see what? It pushes into the cell membrane, like little fingers. And then what it does is it gets through the cell wall and it robs the nutrients from the plant cell. Farmers many times have problems with these doing problems to their crops. But now, another very important thing, so maybe this is the fourth, I don't remember, but a very important thing. Without fungus, we wouldn't be here. Isn't that cool? Without a special type of fungus known as this word, mycorrhizae, engaging in this thing we call a mutualistic relationship, 90% of all vascular plants, 90% of all plants that feed us would not be here. All right? 90% of all vascular plants on their roots, if you dug them up, you'd see a picture that looks on the right. Looks like a fuzzy root. The root is the plant. The fuzziness is the mycelial of the fungus that grows on it, the mycorrhizae. The mycorrhizae gets a place to live. The plant roots provide it with carbon so it can build. There's plus. And guess what the mycorrhizae do? The mycorrhizae digest those nutrients in the soil. And of course, now plants' roots are good for absorbing and anchoring. So once that big old pool of nice, rich nutrients has been dissolved, the fungi use what they need. And just like plants growing for us so we can eat it, the excess is stored in the soil or kept in the soil until plants come along and suck that stuff up. Look at the picture up above. Beautiful scientific experiment. The plant on the left, they spread it with a fungicide, something to kill fungus, killed the mycorrhizae, didn't kill the plant. On the right, they left it alone. That was the control. Look at the difference in growth. It's obvious, right? All right, I got one more minute to explain to you one of my most fascinating things. You can, at the bottom, what the mycorrhizae do. It extends the root system, increases the surface area, increases absorption. But I want to talk about Indian pipes. Indian pipes, Monotropa uniflora, grow on campus. I know where they grow on campus. Unfortunately, I went back over there a couple days ago, and they're not growing right now. They generally are better in the fall, but it's a flowering plant. Okay, now we just finished talking about flowering plants, but generally see, it's got little petals. I know it's 9:20, stick with me for just 1 second though, but it's not green. So plant, regardless of whether you're flowering or not, immediately not being green, you're like, how does it survive? It doesn't photosynthesize at all. This is an incredible adaptation of Mother Nature. This plant has figured out how to be a flowering plant with the stamen and the and basically it does not have to do a lick of work. It doesn't photosynthesize. Now, what's even more interesting about this is how does it live? Well, it basically parasitizes the fungus, the mycorrhizae. It actually grows into it with its roots and extracts some of the nutrients from it that are living on the root of the plants. Three things involved: the Indian pipe, the mycorrhizae, and the plant. A tripartite relationship. I wrote it in red. Isn't that cool? Life always finds a way, doesn't it? These organisms don't photosynthesize. They live on an organism that lives on another organism. Now, the mycorrhizae don't parasitize the plant root, but I believe the Indian pipe does. So there's damage done to the mycorrhizae. It might be mutualistic as well, though, or at least commensalistic. Isn't that cool? All right, hopefully they'll come up this semester and I can show you these Indian pipe. They're really neat plants. I can show you the dead ones, but ain't much to them. All right, I'm going to stop the recording.